Good morning. So today our discussion topic is nothing but a bacteriological analysis of the water. So why we need to do bacteriological analysis of water? So is there any importance, especially why we have to perform bacteriological analysis of water, biological analysis of water? Both are same thing. Why we need to perform this practical? Because our major body composition is nothing but water, and our daily consumption of water is more than the solid. Are you agree this point? So our major consumption is nothing but the water only. So we need to ensure that the water what we drinking, it has to be completely free from pathogenic microorganisms, especially pathogenic microorganisms. So what are the water sources? What are the water sources? It might be a river water, well water, or the rainwater, or yes, supplied from the municipality. So whatever may be the water source, we need to ensure that the particular water what we are consuming is free from pathogens. First, for that reason only in our lab we are going to perform or checking the water what we are consuming is free from pathogen or not. The aim of the practical is nothing but enumerate the coliform bacteria. So coliform bacteria is the main pathogen present in the waters, fecal or whatever the contamination present in the water, especially the coliform bacteria achieves growth inside the water, and it will go inside and it produce discomfort or allergy towards our digestive system. Thereby it might be produced a diarrhea, dysentery or any life-threatening disease according to the immunity or vulnerable populations. You know, vulnerable population is nothing but geriatric and pediatric. Next, what is the principle behind this bacteriological analysis of water? So in our lab, especially we are going to identify the gram-negative coliform bacteria as well as the E. coli. How we can identify? By doing the experiment. Okay, later we will discuss about the procedure. By doing the experiment, there is an indirect methodology. By observe that methodology, we can identify the presence of the bacteria. So if the bacteria comes under the category of the coliform bacteria, it has the ability to ferment the sugar by giving the byproducts of gas as well as the acid. That is the reason why if you have any E. coli infection in our body, we may produce gas accumulation in our stomach and acidity. Did you observe these symptoms? Any of you, have you ever been observed these type of symptoms in your body? Gas production or any acidity, that indicates that the food or water source, whatever may be, it might be contaminated by the E. coli or the coliform producing bacteria. Because if the bacteria is comes under the category of the coliform, sure it will ferment the sugar inside our body or inside our digestive system. Thereby it produces the more accumulation of the gas bubbles as well as the acid. Next, the method what we are going to prefer for these practical is nothing but an MPN method, the most probable numbering method or most probable number method. Okay, so here the method is nothing but very simple. Usually we take the broth. You know, the broth is nothing but a liquid culture of the microbes. Agar is the solid culture. Here we take 3 sets of the test tubes. Okay, 3 sets test tubes contains total 15 test tubes. Each set contains 5 test tubes. The first test tube contains 5 ml of double strength of broth plus 50 ml of water. The second set of the 5 test tubes contain 10 ml of water and single strength of the broth. Means half of the quantity of the broth when compared to the first set of 5 test tubes. The third set of 5 test tubes contains the same single strength of the broth as well as 1 ml of the water. This is the methodology. This is the method we have to perform the most probable numbering method. So broth is there. Inside we pour the water what we are going to test. Okay. First set of test tubes contains 50 ml of water with double strength of the broth. It's nothing but a nutrient medium of the particular bacteria. Second test of 5 test tubes contains 10 ml of water, 5 ml of single strength of broth, and third set of test tubes contain 1 ml of water with single strength of nutrient broth. Next, as usual, it will go for the incubation. After did the experiment, it will go for the incubation up to the range of 24 to 48 hours. The range of 24 to 48 hours. Next, observation or interpretation of the results. So there are two interpretations we can perform. The first interpretation is nothing but for the MPN method, most probable number method. That is nothing but total coliform count. Comes under the category of the total coliform count, not for E. coli. E. coli later we will discuss. So there is a special tubes. It's called as Durham's tubes. It will be inserted into the all test tubes in the inverted position. If the presence of the coliform bacteria or it achieves the growth inside the broth means it releases the gas bubbles. The gas bubbles will go and reach the top of the inverted Durham tubes and it will be floating on the surface of the broth. You know, gas displaces the materials. So the gas bubbles arising from the coliform's presence reach the top of the inverted position of the Durham tubes. Thereby, it will float in half of the manner of the nutrient broth test tube. This is the indication, indirect indication of the gas producing bacteria or coliform bacteria present in the particular sample. Okay, if there is no gas bubble or the Durham tubes is completely deep inside the nutrient broth means it indicates that there is no growth. Next, E. coli conformation. So especially for E. coli conformation, along with the MPN gas production test or displacement of the Durham's tubes, we will perform another biochemical test for identification, especially for E. coli. The simplified pattern or the abbreviation is nothing but IMVIC. I indicates indole production test, M indicates methyl red test, V indicates Vox-Prosker test, and C indicates citrate utilization test. So the pattern of the test, okay, positive for the indole and methyl red confirms the coliform bacteria, and negative for the Vox-Prosker and citrate utilization is nothing but the presence of the coliform bacteria. So the pattern of the IMVIC is nothing but positive, positive, negative and negative. But this is the thing we are going to perform. So once identification the MPN number, then we will go for the E. coli conformation. There is a two set of practicals or two set of combination. So how to understand?